All right, so today we're going to jump back in time a bit to the 1980s to look at this super clever piece of bioengineering called Lambda ZAP. It was a total game changer for scientists hunting for genes. But to really get why it was so cool, you first have to understand the massive, massive problem they were up against.
So just imagine this for a second. Your entire genome is like a library with millions of books, and your job is to find one specific sentence hidden somewhere inside. That's pretty much what geneticists were dealing with, trying to pull a single gene out of an organism's entire DNA. I mean, how do you even start to do that efficiently? Back in the early days of genetic engineering, this wasn't just some abstract puzzle, it was a real-world bottleneck. It was slowing everything down. Scientists had gotten pretty good at making these huge libraries of genes, but actually getting their hands on the one they wanted to study, that was incredibly slow, tedious work. And here's the pickle they were in. You had two main tools, and they were kind of polar opposites. On one side, you had the Lambda phage vector. Think of this thing as a giant cargo plane. It's fantastic for carrying a huge, diverse library of genes into bacteria, perfect for that initial search. But once you find your gene, the Lambda vector is massive and clunky, a real pain to analyze in detail. On the other side, you had the plasmid vector. This is like a nimble little courier van. It's small, simple, and absolutely perfect for detailed work like sequencing. The catch? It's really inefficient for creating that big library in the first place. So you were stuck. And hey, don't just take my word for it. The inventors of Lambda ZAP themselves said it perfectly in their paper. They talked about the process of moving a gene from the big vector to the small one as being lengthy and time-consuming. You could almost feel the frustration. This was the exact bottleneck they wanted to smash. So how'd they do it? Well, a team at a company called Stratagene came up with an idea that was honestly just brilliantly simple. They created something they called a vector in a vector. The whole idea was to just stop thinking of these two tools as separate things. Instead, they asked a really powerful question. What if we could combine their strengths? What if we could literally build that nimble courier van directly inside the giant cargo plane? And this right here is the actual blueprint from their 1988 paper. You can see exactly what they did. They took the DNA of that small, useful plasmid, P bluescript in this case, and they just spliced it right into the middle of the much, much larger Lambda phage DNA. They literally built the van inside the plane. It's awesome. So the big idea breaks down into these three key steps. First, you use the power of the Lambda phage to do the heavy lifting. Create your gene library and find your target. Second, that gene you just found is already sitting inside the easy-to-use plasmid. And third, and this is the real magic trick, they engineered a biological eject button to get that plasmid out completely automatically. Yeah, this eject button, that was the absolute key to the whole thing. The technical term is in vivo excision, which is just a fancy way of saying the plasmid cuts itself out while it's still inside a living bacterial cell. And how it pulls that off, that's where things get really ingenious. This diagram from the paper lays out the whole process perfectly. A scientist finds the Lambda phage with their gene of interest. That's on the left. They use it to infect a bacterial cell, but they also add a second, very special virus at the same time, and that is what pulls the trigger on the whole automatic process. That special virus is called a helper phage, and it has basically one job: to show up with the molecular scissors, the proteins, that are needed to snip the plasmid out. You can think of it like a biological key that unlocks the plasmid from that big Lambda phage shell. Okay, so let's walk through it. Step 1, you infect the cell with both the Lambda ZAP vector and the helper phage. Step 2, proteins from that helper phage go hunting for these special cut-here signals that the scientist engineered right onto the DNA on either side of the plasmid. Step 3, those proteins start copying a single strand of the plasmid's DNA. And finally, step 4, they snip that new strand free, curl it into a circle, and package it up, all ready for the scientist to collect. It's all done for you, no more manual cutting and pasting. So, that's the theory. It sounds amazing, right? But the biggest question for any new invention is always, does it actually work in the real world? The team at Stratagene had to prove that their clever system wasn't just a great idea on paper. So they did what any good scientist would do. They set up a real-world test. They used their new Lambda ZAP vector to create a gene library from chicken liver cells. And then they went hunting for a gene they already knew a lot about, the actin gene, to see if they could not only find it, but more importantly, if they could actually get it out using their new automatic system. And the results were just incredible. This is data straight from their paper, showing what happened when they hit that eject button. Just look at that last column, total colonies recovered. It didn't matter if the gene insert was small, like 1,400 base pairs, or pretty big, over 3,000. The system worked like a charm every time, creating millions of bacterial colonies, each one a tiny factory cranking out the neatly excised plasmid.
I mean, let's just sit with that number for a second. Over 1 million. From one single successful find, one little plaque on a petri dish, they could get more than 1 million copies of the plasmid ready to go for analysis. This wasn't just a small improvement. This was a giant leap in efficiency.
And here's the final visual proof. They needed to make sure their new system was just as good at finding the gene as the old school method, which was called Lambda ZAP 11. On the left, in image A, you see these nice, clear positive spots from Lambda ZAP. On the right, in B, is the old way. The signals are just as strong. So the bottom line is, Lambda ZAP was just as effective at finding the needle in the haystack, but it came with that absolute game-changing automatic excision feature.
So when you put it all together, what was the real impact of this thing? Why are we even talking about it today? Well, it really kicked off a revolution in the speed and efficiency of genetics research. I think the authors themselves summed it up best right in their abstract. Their whole goal was to significantly increase the rate at which clones can be isolated and analyzed. And boy, did they succeed.
So let's just recap the huge advantage here. By cleverly packaging the simple plasmid inside the powerful lambda phage, Lambda ZAP just completely got rid of that slow, painful manual step of subcloning. I mean, this single invention saved researchers an unbelievable amount of time and effort, and it just massively sped up the whole pace of gene discovery.
You really can't overstate how big a deal this was. Tools like Lambda ZAP were the engines that powered the whole genetic revolution of the late 80s and 90s. It helped make huge projects like the Human Genome Project even conceivable, because it made that fundamental first step, going from a giant library to a single gene, faster and easier than ever before. Which kind of leaves us with this final thought. Back in 1988, Lambda ZAP came along and just zapped a major bottleneck, unleashing this huge wave of discovery. So it really makes you wonder, what are the big bottlenecks holding back science right now? And who are the brilliant bioengineers out there today, building the clever new tools that are going to break them wide open?